I'd like to introduce next Dr. Chester Whitley. He's a tenured professor in the Department of Pediatrics, Experimental Clinical Pharmacology, and director of the Gene Therapy Center and Advanced Therapies Program at the University of Minnesota. But I also wanted to share with you, he's the founding course director of the annual We're Organizing Research on Lysosomal Diseases research meeting, which we all have coined as the word world, one of the largest meetings in the world for lysosomal diseases. Recently, Dr. Whitley and his colleagues evaluated the zinc finger nuclease technology for Sangamo Therapeutics. This led to the first Union clinical trial of the in vivo gene editing form of gene therapy, for which the first human subject was treated in phase 1 clinical trials for MPS 1 and MPS 2. He's going to talk about navigating complexities of clinical trials before we have industry updates. Let's welcome Dr. Chester Whitley.
Thank you, Terry, and thank you, everybody, for coming. Because we're running a little late, I'm going to accelerate this a little bit. It's a bit of an experiment here. I've got some new slides that I've really not tried out, and I hope you'll find this interesting. Of course, I'm involved with very many private entities, including the NIH Lysosomal Disease Network, for which we've recently been refunded for the next 5 years. Thanks to many of you for your encouragement and support. I'll talk about that only briefly. It'll be intertwined in the things that I talk about. The National Institutes of Health was mandated by Congress, by actual legislation, something like 20 years ago, because this group lobbied their local Congress representatives state by state. And that legislation required the National Institutes of Health to fund clinical research, in other words, involving human beings, not animals, in networked research across the country to look at orphan or rare diseases. And this is where the program has gone now. They funded initially 3 different rare disease consortia, and now they're funding 20. These are the lysosomal diseases. We try to include all of them, and I'll allude to how we do that in the near future here as we proceed on. But I want to talk about the new lysosomal disease network, what we're doing slightly differently by NIH mandate or requirement. I'll talk a little bit about the history and themes and structure, but I won't spend a lot of time talking about this because what you all came here to hear was about clinical trials and navigating clinical trials. This is the history, however. You'll see across the top, in 2003, we applied for that NIH funding mechanism. We did not get that NIH funding mechanism. There were 3 networks, including one organized by Joe Munzer, and the pink sheets at the end of the review said, you 3 networks interested in lysosomal diseases are great. You're the dream team. That was the word they used, but you got to work together. So therefore, they did fund meetings annually, and for 4 or 5 years, we had annual meetings, which became the World Symposia. After that point, we were in a position to for another application, and we applied. But during that 5 years of no lysosomal disease network, we also kept the World Symposium going. And as you'll see right here, the registrations have increased. In February, back here in Orlando, will be another World Symposium, and we anticipate over 2,000 people to attend. You're all welcome. It's intended predominantly for scientists, for clinical researchers, for pharmaceutical companies, but everybody interested in lysosomal diseases, including patients and relatives, are certainly more than welcome. The NIH funds this lysosomal disease network by a mechanism called the RDC, or Rare Disease Clinical Research Network. And last night I got an announcement they're going to have a new logo, but their logo was in the center, those yellow swirly things. But you can see they're funding the lysosomal disease network now, as indicated here in white, but also things like brittle bone disease and urea cycle disorders. And they are requiring us to be very intimately involved with patient advocate groups. In fact, each one of these yellow 20 rare disease consortia, they typically have one patient advocate group they're involved with. Well, because there are so many lysosomal diseases, and we'd all like to play together, there are actually 65 or more patient advocate groups we're trying to be involved with, and we've been very successful. And certainly the MPS Society, you have been very integral in our success, if you will, and helping us move forward. The data is all submitted to a DMCC over here on the right, the Data Management and Coordinating Center. And so the data will become public knowledge as it evolves. That's how the mechanism works. Well, we're talking about finding clinical trials, and I think, let me ask, how many people know about clinicaltrials.gov? I hope everybody knows, but I'm going to talk about this a couple of times because this changes day by day, month by month. I'm not going to show you any screenshots of it, but if you go there, you'll see what happens. You can put in the name of your condition that you're interested in. You can put in the name of a company. You can put in even a symptom sometimes, and you'll see clinical trials that might be interesting to you. At the bottom, it will typically list the contact information, typically a phone number for somebody to call to see if you're eligible or see how you get involved. As a background, I want to give you some general ideas about clinical trials. Some of this may be very, very well known to all of you. Those of us who run clinical trials have had to go through this process. I want to thank John, whose slides I borrowed for this presentation and modified slightly. But this is what a clinical trial is in background, the process, an overview. So when we do a clinical trial, it's typically to test a new treatment, either a drug or a device. But we're looking at a specific disease, we're assessing the safety of that drug or treatment or device, and we're defining things such as the route of administration. Is it IV or is it oral or some other route? We're defining the dose. We're testing it for safety. We're exploring combination therapies. And we're looking in the long term, we hope, at quality of life. But frankly, that takes a very, very long term to assess in many cases. Sometimes we're looking at prevention of a disease or early detection or even a diagnostic test. Typically, any clinical trial runs through 4 phases. This is globally recognized. Phase 1 is where we test the drug for safety. What side effects does it cause? And frankly, there is no drug or no device that I know of that has absolutely no side effects. But we try to define that in a small number of patients. Now you'll see here under phase 1, this slideshow says, well, 15 to 30 patients would be a good number. I'll tell you, that could be the whole population of lysosomal diseases in the US for some conditions. It's that rare. We're talking about orphan diseases. Phase 2 moves on to start assessing efficacy. There you want more than 100 patients, typically, or close to it. By the time you get to phase 3, where you're really looking systematically at the treatment, the efficacy and the safety, you're into testing thousands of patients. And phase 4, where you really look at what happens with the drug after it's already approved by the FDA or the European Medicines Association, can involve many tens of thousands in some cases. Who are the people involved? Well, obviously, we as human subjects, and I've been a human subject in more than one clinical trial myself. We're looking at the physicians who are your interface with the sponsor or the manufacturer. Lots of nurses, you all know them and love them well. Pharmacists, lab techs, social workers, and data managers. There's a big group of people behind a study, which is why it costs so much money in many cases. The research protocol, the roadmap. Well, when we're writing something up as a clinical trial, these are some of the things that we have to consider. Documenting what are the objectives, the background, the rationale, the treatment plan, the procedures, if any, that will be done. Eventually, how do we evaluate the response? And how do we analyze that statistically so we know we're not just being fooled by what looks like a good number, but we're actually testing that drug in a statistically significant manner. There is huge federal regulations around how we do clinical trials. These are just some of the items kind of outlined here. They're all in the Code of Federal Regulations, what's called Title 21 or 21. It's hundreds and hundreds of pages long that defines how one does a clinical trial under federal regulations. What about international regulation? Well, there are internationally applicable, typically ethical, but also regulatory procedural rules that we also are obeying. Most drugs nowadays are tested globally, not just in the US. If they're started outside of the US, they want to come to the US because the FDA is considered the gold standard. If they're tested initially in the FDA, that's a great launch pad for using the rest of the globe to extend the use of that drug. And why is it so highly regulated? Well, there have been passed what they call transgressions of human ethical and statistical and other aspects. The Nuremberg doctors during World War II were not practicing good medicine as they were doing unethical research. There was the thalidomide tragedy. There's the Tuskegee experiments where blacks were discriminated. There are human radiation experiments, and more recently we have gene transfer experiments where the ethics of those people involved or the conduct of the study, even well-intended, was not up to what we could be doing. So this is the history of why we have such a dense regulatory environment to work in. Informed consent is the central part of this. That when you're consented into a study, the person who you're signing the consent form with has to know what's going on, has to answer all your questions, and make sure you really understand as best the two of you can understand that. Basically, it has to be voluntary. It's not coerced. The Institutional Review Board, or the IRB, or sometimes called the Ethics Committee, supervises all we do, particularly in determining who is eligible and did that person get complete and fair informed consent. The challenge is often recruitment. How do you find enough patients who are interested in participating in the research to make it happen? Notice bullet point 2, only 3 to 5% of newly diagnosed cancer patients actually participate in a clinical trial. And in areas where we're still learning a lot, and where a lot of the first use of a for the first use of a treatment has to be in the setting of a clinical trial, that's a small number. Wouldn't it be great if 100% of people who are eligible did participate? But finding those people is part of the battle. Convincing people that it's worthwhile for them is another part of the battle, and sometimes they view it as not worthwhile. But that's the challenge of getting enough numbers to prove a treatment does or does not work, is safe, or has excessive side effects. Getting enough people to come up to that statistically significant assessment is a challenge. So what are the recruitment strategies? I just gave you the best one, didn't I? www.clinicaltrials.gov. But physician staffs, newspaper ads are sometimes employed, and sometimes the physician in your own clinic might be part of a clinical trial, and they may approach you as well. But here's the one that's the gold standard, the one that's accessible to everybody. Go to clinicaltrials.gov. So once the data are collected, there's some things that are also happening. You don't see them all, but the nurses and technical staff, the physicians, are recording data on case report forms. More and more of that is becoming not paper, but electronic. It has to be all preserved and safe and reproducible and findable in the future. Any kind of serious adverse effect has to be recorded. That's why we have different levels of scrutiny about what you report or what we observe when a clinical trial is happening. Importantly, what's the clinical trial end product? Why are we bothering to do this? Well, the ideal is unambiguous conclusions regarding the clinical outcome and of the test, treatment, or device. That's the ideal. That's why we bother to spend tens of millions of dollars to do clinical trials. What really happens? Well, we strive for the ideal, but in many cases, we have to settle for the best compromise. But at least the information is there. The FDA, European Medicines Regulations Agency, patients, physicians, we look at that outcome. It's often on the label of the drug or the device. So if you get that little package insert, you should be aware that's worthwhile reading if you have questions about a drug or a treatment. That is a legal document negotiated by the FDA and the sponsor, the company making the product. They cannot change a single word of that unless both parties agree to it. So look at that. It lists the safety, in other words, what are the side effects? Sometimes I kind of joke and say, well, what if there's a lot of automobile accidents to various people during the clinical trial? Does that appear on the label? Well, yeah, it can. I don't think it ever has been, but coughs and colds and fevers, how many times does somebody encounter something like that during the clinical trial? That gets recorded. It gets reviewed by the company and the FDA to say, well, was it really possibly related to the drug or not? But it might be important. So if maybe you're losing your vision because of a drug and there are a lot of automobile accidents during the clinical trial, well, maybe there's some information there we should understand that we didn't think about. How does that work in our world? How does that work in the real world of new therapies for lysosomal diseases and specifically mucopolysaccharidosis? So I want to pull up one or two examples here quickly. I want to talk about clinical trials or the background work in animals, the things that are done before a human being actually gets contact with the drug. That's called preclinical. Phase 1 I mentioned was the safety. Phase 2 is looking at efficacy, if you will. Phase 3 is a larger number of people undergoing treatment. And then what happens after a drug or device is actually approved by the FDA? There's still FDA ongoing oversight. If any of you are involved with a Genzyme Sanofi product, you may or may not have signed up for the Genzyme or Sanofi disease registries. That's required by the FDA, of the company. It's not complete because many of you did not sign up for the post-marketing study, but it's the required mechanism that Genzyme Sanofi has used to fulfill their commitment to the FDA, to at least make some attempt to look at additional side effects and efficacy and other issues after the drug is commercially available for sale. So, how about our real world of lysosomal diseases? Well, arguably, and I don't think there's any exception to this, bone marrow transplant for Hurler syndrome was the very first successful systemic therapy for any lysosomal disease. So let's look at the preclinical studies, the phase 1, the phase 2, the phase 3, the post-marketing regulatory process. The reality is, there was none. Bone marrow transplant is not a drug, it is not a device. It's a surgical procedure, if you will, or a clinical procedure. So there is no FDA oversight. What we do know about bone marrow transplant for Hurler syndrome is kind of like this. Back in England, and Paul Orchard alluded to this, Jack Hobbs treated one child with Hurler syndrome doing a bone marrow transplant. It was reported in the Lancet, a very credible journal. It had peer review by probably at least 2 or 3 other scientists, physicians. And as I tell this story, and I hope this is not, that Jack Hobbs is okay with this, nobody believed it. How do you get enzyme from the bone marrow into the blood and importantly across the blood-brain barrier? Impossible. It just does not seem likely. So Bill Crivet, University of Minnesota, was always trying to do things on the cutting edge. He got the reputation, Wild Bill. Thank you, Bill, for being so wild. And he could not convince our University of Minnesota bone marrow transplant colleagues to do the first transplant when we knew the blood-brain barrier existed. So he got them to say, well, for MPS 6, let's try it for MPS 6. And they transplanted a young 18-year-old woman who had very, very bad respiratory problems. She survived the transplant. She was fully grafted. They published it in the Lancet. Not the Lancet, sorry, the New England Journal of Medicine. Well, that was enough to say to the other bone marrow transplant people who are oncologists that we can do it. And Terry is telling me I'm out of time. So at that, I'll say we did the first transplant in the US, and it took us 5 to 10 years to know that we could prevent brain disease. So I'll be happy to show you the other half of these slides sometime. Thanks very much, everybody. Thank you, Terry.